Look closely at a crust of sourdough bread, a block of aged cheddar, or a roasted coffee bean. To get these prized flavors and textures, we deliberately subject the raw ingredients to a process of controlled decay. Normally, when organic matter breaks down, a chaotic mix of environmental microbes tears it apart, leaving it toxic and rotting. But under the right conditions, a highly specific set of organisms takes over. We call this precision breakdown fermentation. Biologically, fermentation is a metabolic process. It happens when microorganisms extract energy from carbohydrates, like the sugars in milk or wheat, in environments completely cut off from oxygen. Mastering this biological plan B gave early humans a way to halt unpredictable rotting. By steering the decay toward a predictable outcome, they secured calories that would otherwise be lost to spoilage. Under normal conditions, neon blue oxygen flows into a cell, burning sugar to produce white sparks of ATP, adenosine triphosphate. When oxygen is cut off, the cell dims. To survive, it smashes a gold sugar molecule to keep ATP sparking. This specific chemical hack is called anaerobic respiration. It bypasses the need for oxygen entirely, allowing the organism to pull just enough energy from its surroundings to stay alive. Because the cell is ripping apart sugars without oxygen, it leaves behind unburned, energy-rich chemical byproducts. Those microscopic waste products eventually become the very substances that shaped human diets and economies. For thousands of years, the mechanics generating those byproducts were completely invisible. Ancient brewers and bakers watched vats of grain bubble and seethe, assuming it was a spontaneous chemical reaction. Even the word yeast traces its roots back to ancient words meaning to boil. That mystery held until the mid-19th century. The French scientist pictured here, Louis Pasteur, proved that the bubbling vats were actually teeming with microscopic living organisms that were actively initiating the chemical changes. A few decades later, the German chemist seen in this portrait, Eduard Buchner, ground up those microbes and extracted a liquid juice. He proved that non-living enzymes inside the organisms were doing the actual work of fermentation, demonstrating that life's most complex chemical changes could be isolated, studied, and measured outside of a living cell. We now know the primary engine Buchner and Pasteur were studying is Saccharomyces cerevisiae, a single-celled fungus commonly known as yeast. When placed in a sugary environment without oxygen, yeast relies on a specific survival pathway called ethanol fermentation. Watch how this mechanical breakdown occurs. The glowing white yeast cell consumes the sugar sphere, extracting its tiny fraction of ATP. It then violently ejects the leftover molecular waste, a clear liquid ethanol, and a rapidly expanding bubble of carbon dioxide gas. When you trap millions of these expanding carbon dioxide bubbles inside a matrix of wet flour, they stretch the elastic gluten webs outward. That biological waste gas provides the internal architecture and physical lift required to bake a loaf of bread. Yeast is a highly efficient engine, but it is only one type of survival mechanism. When we move from fungi to bacteria, we find a different metabolic pathway altogether, lactic acid fermentation. In this diagram, you can see a cluster of geometric neon blue bacteria. When they consume sugars in an environment devoid of oxygen, they do not produce alcohol. Instead, they pump out lactic acid, forming a dark, murky cloud. This drops the local pH rapidly. When competing, jagged green pathogenic microbes enter the space. The harsh acidity breaches their cell walls, dissolving the invader on contact, creating a toxic biological force field. Humans learn to trigger this microscopic turf war. By submerging vegetables in salt water or keeping milk in warm containers, they created the perfect oxygen-free environments for lactic acid bacteria to thrive and deploy their acid shields. The resulting sauerkraut and yogurt lasted for months without spoiling. Early fermentation was a method of hijacking a bacterial defense mechanism to ward off the wild microbes that caused food to rot. Scaling up from these cellular mechanics reveals a massive ecological advantage. Manipulating microbial waste allowed early societies to exert control over their caloric environment. A raw harvest of wheat, grapes, or milk has a strict biological expiration date. But by filtering those calories through yeast and bacteria, societies produced stable, nutrient-dense foods that could be stored safely through long, freezing winters or extensive droughts. You cannot build a dense urban center if your food supply spoils in a week. The caloric stability provided by vats of fermented grain and cellars of preserved dairy allowed populations to cluster together in the first permanent cities. We easily recognize how human history was shaped by the visible domestication of wheat, dogs, and cattle. We fenced in pastures and selected seeds to ensure our physical survival. But long before we understood the biology, we were setting traps for wild microbes and selecting the strains that produced the best chemical shields. Human civilization relied entirely on domesticating the invisible world.